Section 8.4, Transcription. So RNA, or ribonucleic acid, carries the instructions from DNA in one direction. Okay, it goes from DNA to RNA to protein synthesis. Okay, this is called the central dogma of protein synthesis, where DNA has the instructions, RNA carries the instructions, and uses the message from DNA to make the proteins. Replication is the process of copying DNA, and transcription is writing down the message in DNA into RNA language. Translation interprets the DNA message into a string of amino acids, okay, which would make up the protein. Okay, so DNA in the nucleus here is going to undergo transcription, where just part of the DNA molecule, just the gene that codes for a specific protein, will be copied into RNA language in terms of messenger RNA. The messenger RNA will leave the nucleus and go to the ribosome, and then the message on this messenger RNA will be translated into amino acids, or a chain of amino acids that are linked together that will eventually become the protein. In prokaryotes, remember prokaryotes are bacteria, much simpler, much less complex than eukaryotes. Replication, which is the copying of DNA. Transcription, which is taking part of the DNA and making it into a messenger RNA. And translation, which is the actual making of the proteins, occur in the cytoplasm at roughly the same time. This is all kind of going on at the same time. Remember, the life cycle of bacteria, or a prokaryote, is very, very quick, so it's going to have to be doing things kind of simultaneously to handle all its tasks that it needs to take care of. In eukaryotes, since much more complex, the processes are separated, meaning replication, transcription, and translation are separated, and not just location, meaning where it's happening, but when it's happening. Okay? Replication, remember, replication is the copying of DNA, and it's going to copy DNA before the cell is going to undergo cell division. Okay, so DNA has to replicate before cell division. Okay, transcription happens anytime the cell would need a protein for a specific task, and just that one part of the DNA molecule would open up and get copied into messenger RNA through this process of transcription. Okay, both these guys take place in the nucleus, replication and transcription. Before this messenger RNA leaves the nucleus, it has to undergo a little bit of a processing process, which we're going to talk about in a bit, where it gets modified and gets it a much more exact message. Then the messenger RNA will leave the nucleus, and it'll go to the cytoplasm, specifically the ribosome, where this message from messenger RNA will be translated into an amino acid sequence, which will become the protein. So a few examples of when gene expression occurs. One would be Arctic fox fur color change. So remember, in the winter, Arctic fox is going to be white. It's not going to be producing the proteins that would make for pigment in the fur. The reason it would do that is so it will blend into its environment a little bit better. In the winter, there's a lot of snow, so it's going to blend in. It'll make itself a better predator that way, so it can sneak up on things, and also can hide from larger predators so it doesn't get eaten. In the spring and summer, it's going to have pigment produced, because those proteins necessary to make coat color or fur color in the Arctic fox will start being made. So again, it can blend into its environment a little bit better. In the spring, summer, early fall, there's not going to be snow, so it's going to be kind of a brownish environment. It's going to blend in better. Okay, start of puberty. Okay, that is a bunch of different genes getting turned on that will produce all the secondary sex characteristics: facial hair, pubic hair, muscle mass, menstruation in women, production of sperm in males. All this stuff is happening through these genes getting turned on in your DNA and being expressed. And healing a cut. Anytime you need to heal a cut, okay, she's got a little boo-boo on her knee, fell down, went boom. You need to produce some proteins that are going to help with the clotting process and help it scab, the clotting or the scabbing up of that. Keeps the body from losing materials, bleeding out anymore, but it also keeps bad stuff, bacteria and whatnot, from getting into the skin, because your skin is a barrier to protect you, not just to keep stuff in, but also to keep stuff out. And so it makes a protective layer with this scab. So comparing DNA and RNA, remember they're both nucleic acids, so they both fall into the same general category of biological molecules, nucleic acids. Okay, but they are not the same. Okay, they are similar, but they're not the same. If you look at these guys side by side, first DNA is a double strand. Okay, it is a double helix. It is two strands of nucleotides linked together, where RNA is a single strand. Okay, DNA is going to be a lot bigger. It's going to carry a lot more information. On one DNA molecule, you could have several genes. On a messenger RNA, it's going to be just the copy of one gene. So it'll be much shorter, much less information, because it's coding for a specific gene rather than coding for a whole bunch of genes. Okay, so they are both similar in that they are both chains of nucleotides. Remember, a nucleotide is a phosphate, a sugar, and a nitrogen-containing base. Okay, the sugar in RNA is ribose, and the sugar in DNA is deoxyribose. Okay, in RNA, thymine is replaced with uracil. Okay, remember the four we talked about in DNA were adenine, thymine, guanine, and cytosine. Okay, so thymine would be in DNA, but in RNA, there is no thymine. It gets replaced with this other nucleotide called uracil. Okay, and DNA is double-stranded. RNA is single-stranded. Okay, there are others, but not so important for our purposes. DNA is found in the nucleus. RNA is made in the nucleus and then leaves. Yeah, DNA is a lot bigger. There's many different copies of DNA. DNA is going to replicate every time the cell divides. RNA is going to be made anytime the cell needs a protein to be made. So there are some other differences, but these are like the big ones that you need to be responsible for. Okay, so for the base pairing rules for RNA, remember RNA is coded for from DNA. So RNA is going to get its instructions from DNA. So on DNA, if there's an adenine, the complementary base that's going to form on RNA is uracil. If it's a thymine, it would be adenine. Guanine on RNA would give cytosine, and cysteine would give guanine, or cytosine would give guanine. Okay, so DNA has 4 different bases in RNA, and the base pairing rules are the same as Chargaff's rules for DNA, except with thymine being replaced with uracil. Okay, another difference between DNA and RNA is there are 3 different types of RNA. The first is called messenger RNA, or mRNA. This is what's formed during transcription, when the DNA opens up and it's making a copy of that specific gene to code for a protein. Okay, so the messenger RNA transcript, the thing that's formed from the DNA strand, would be complementary to the DNA strand. So if DNA is ATG, according to the base pairing rules, this would be UAC on our messenger RNA, right? Messenger RNA gives that. CCT on DNA would give GA on RNA. TAG on DNA would give AUC on messenger RNA, and CTA would give us GAU. So this would be the messenger RNA transcript, or the messenger RNA complementary strand to the DNA that was reading it. Okay, so that's one type, messenger RNA. Okay, the second type is ribosomal RNA, or rRNA. rRNA forms parts of the ribosomes, like the two major subunits that make up the ribosome are formed from ribosomal RNA. Remember, the ribosome's function is to make proteins. And the last type of RNA is called transfer RNA, or tRNA. And what this does is it brings the appropriate amino acid from the cytoplasm to the ribosomes to make the growing protein chain, or there's a growing strand of amino acids that eventually turns into the protein. Okay, so messenger or RNA, there's three different types. Okay, messenger RNA, rRNA, tRNA. They have very different functions. They have different looks, but they all work together for one thing, which is to make proteins. That is their job. So how does messenger RNA get formed? Well, it makes its form through this process of transcription. Okay, transcription is the copying of a sequence of DNA, and the DNA we're copying will be a gene. Remember, a gene is going to code for a specific trait, and this sequence of DNA will produce a complementary strand of RNA. This complementary RNA strand would be in the form of messenger RNA. Okay, so there's an enzyme that controls this, and this process is catalyzed, being it happens through this enzyme called RNA polymerase. Okay, it does a couple things. It opens up the DNA helix, and it also bonds the nucleotides. Now these would be that RNA nucleotides together in a chain to make RNA. Okay. You can see this. This is a DNA molecule here. RNA polymerase is going to hit, and it's going to find the front of the gene. It's going to open up the helix. So these are now unpaired bases on the DNA strand. And then RNA polymerase will bring in the complementary RNA base that will make this strand of messenger RNA, which eventually will get processed and then leave the nucleus and go to the ribosome to make proteins. So, more on transcription. RNA polymerase recognizes the start of a gene and unwinds a segment of the DNA molecule. Okay, so it's opening up the gene, the DNA molecule. RNA polymerase is going to put together complementary RNA nucleotides together using the template. Now the template would be the template strand of the DNA. So it's going to be either one side of this or the other. It's reading one or the other, not both. Okay, and the enzyme will recognize which one is the reading strand, the one that it has to copy, and which one is not. Okay, so the RNA polymerase is going to bring in the appropriate RNA nucleotides. That would be these guys. Okay, according to the base pairing rules. Once it's done that, the DNA is going to close back up. It's going to zip back together and look just like normal. Okay, the messenger RNA strand will detach from DNA, and again, before it leaves the nucleus, it has to get processed, which we're going to talk about later. But there's this thing called messenger RNA processing, which we'll get to in a later section. All this is taking place in the nucleus. Okay, transcription is similar to replication, not the same, but similar. Similarities are it occurs in the nucleus. It's catalyzed by large enzymes. DNA replication is catalyzed by helicases as well as DNA polymerase and others. Transcription is catalyzed by RNA polymerase. Both of these involve unwinding the DNA double helix. For DNA replication, it's gonna form several replication bubbles, so it makes this process replicating go faster. For transcription, it is just opening up just the gene, just one part of it. Okay, both of these are controlled by complementary base pairing rules, and it's highly regulated. Regulated, meaning that it has to happen with the enzymes keeping track of things. The differences are replication is ensuring that each new cell will have one complete set of DNA. Only happens once during the cell cycle, right before the cell is going to undergo mitosis and cell division. Okay, transcription can occur thousands of times during the life cycle of the cell or during the cell cycle. Anytime the cell needs a protein, it can do it. And a bunch of RNA molecules can be transcribed from a single gene. So it can just make copy after copy after copy after copy. Okay, if it needs a whole bunch of proteins at one time, it's going to make a whole bunch of messenger RNA to make a whole bunch of proteins, right? The body will regulate this, the enzymes will read it, and it will happen as needed by the cell.